Hello, and welcome to the ACR Journals on Air podcast. I'm your host, Vicky Shamagam. The most powerful questions are the ones that make people pause, delving into a place of curiosity. And for many rheumatologists, does the sun make you sick is one of those questions. The manuscript we will discuss today is Regulation of Photosensitivity by the Hippo pathway in lupus skin. It was recently published in Arthritis and Rheumatology, and a link to the manuscript and disclosures are in our show notes from today. I'm excited to be joined by the manuscript's senior author, Dr. Michelle Kallenberg. Dr. Kallenberg is the Giles Ball and Dorothy Mulkey Research Professor of Rheumatology and Associate Chief of Basic and Translational Research at the University of Michigan Division of Rheumatology. She completed her MD, PhD, and internal medicine training at Case Western Reserve University and her rheumatology fellowship at the University of Michigan. Her clinical work is focused on caring for complex lupus patients, and her translational research uses patient samples and murine models to uncover mechanisms that drive lupus activity. She is particularly interested in cutaneous lupus, the factors that drive photosensitivity, and understanding how skin inflammation impacts systemic disease in lupus. She's won numerous awards for her research, including the very prestigious Presidential Early Career Award for Scientists and Engineers. Welcome to the show, Michelle.
Thank you so much for having me.
So photosensitivity is one of the hallmarks of lupus, and we know it can be both a debilitating symptom and also drive systemic disease activity. Your lab and others have shown that keratinocytes from non-lesional skin in patients with lupus show increased UVB-driven apoptosis. And you hypothesize that this could be due to epigenetic modification and that signaling through the Hippo pathway may play a role. To get us started, could you fill us in on epigenetic modification? What is it, and what is known about the role of epigenetic modification in lupus in general, and then particularly in the skin?
Thank you for that question. So epigenetic modification are changes to the DNA that change how genes from the DNA can be read. And these are heritable modifications, but they don't actually change the sequence of the DNA, but they can be passed from cell to cell. And the reason that we got interested in it in the skin was because when we take keratinocytes from skin biopsies from patients and we grow them in culture for many weeks, we see changes in those keratinocytes that persist, and they are more pro-inflammatory than healthy control keratinocytes. So for us, that suggested that something was different in those cells that stayed even when the cells were replicating from one generation to the next to the next. And so that's why we looked at epigenetics. In lupus, it's known that there are epigenetic modifications in blood cells, for example, but no one had looked in the skin, and we thought it was an important question to ask.
Yeah, that makes sense. And so let's talk about the hippopotamus in the room. What is the hippo signaling pathway, and why did you want to study it in lupus?
So we didn't want to study it initially. We sort of found it, and you have to go where the science takes you. The hippo signaling has been primarily researched in cancer biology, and also in development signaling and cell adhesion responses. And what is known is that the hippo pathway is very important in terms of being a light switch between whether the cell wants to proliferate or live or die. And so for us, when we hit on the hippo signaling pathway in lupus keratinocytes, that was actually kind of exciting because we know, like you mentioned, that keratinocytes are more prone to dying after UV exposure. And we thought this could be a critical regulator of that pathway.
That's so exciting. I'm imagining you following the hippo through the undergrowth to the watering hole. So in this manuscript, you describe this beautiful series of experiments, each kind of building on the next, starting with methylation profiling. And this experiment aimed to look at epigenetic changes in the keratinocytes from non-lesional skin of lupus patients and compared them to keratinocytes from the same regions from healthy controls. Tell us what you found in that analysis and what was the functional relevance of those methylation findings.
So one of the things that came out from that analysis, sort of on a global scale was that there's really profound differences in how genes are methylated in lupus versus healthy control keratinocytes. And like you said, we were very careful to match our controls by age, sex, and ethnicity to try to make sure that we were controlling for any variables that could possibly influence epigenetic changes. But we still saw there was, increases in methylation and decreases in methylation in lupus versus healthy control keratinocytes. And so when we found the Hippo pathway was the most differentially methylated pathway that came up in the analysis. And when we looked at the various genes that were affected, the one that really stood out to us was that a gene called WWC1 was hypomethylated, suggesting that it would be increase in expression. And WWC1 is a critical on switch for the Hippo pathway, that when it's overexpressed, it's a scaffolding molecule. So it regulates things by just its mere presence. And when it's overexpressed, it tends to turn the Hippo pathway on, which can promote apoptosis.
Fabulous. So then you went on to the next portion of the work that looked at single cell RNA sequencing. Can you explain to our listeners what that is?
So single cell RNA-seq is a really powerful tool that lets us look at gene transcription at a single cell level. So we can look at each keratinocyte, each fibroblast, each T cell that we can get out of a tissue and look exactly what that cell is expressing. So you get a ton of data. So it requires really powerful informatics and really great collaborators to help us analyze that data. But you basically can get a profile of what each single cell in the skin is doing, for example.
And then kind of put it back together into the matrix on the back end with the bioinformatic team. So you specifically looked at the differential expression of the Hippo pathway genes in lupus keratinocytes and compared them to healthy controls. And your graphics in this article are beautiful. You have one in particular that included some violin plots. And because this is an audio podcast, I need you to turn those into music for our listeners' ears. What did you find?
So I'll do my best. So the violin plots, I love your puns. They're wonderful. So what we showed is that we had both data from keratinocytes and from bulk profiling of lesional and skin in lupus patients from various studies we'd done in the past. And we were able to look at genes that are regulated by the Hippo pathway and see whether they were up or down. And the Hippo pathway can get a little confusing because depending on how people talk about it, it's either on or off, but what that means varies in the eye of the beholder. So when I say this, what I mean is that if the Hippo pathway is on, the genes that get regulated by a critical protein called YAP are actually being skewed towards apoptosis. And when YAP is not skewing towards apoptosis, it goes towards proliferation. So these signatures in these cells are looking at the proliferation genes that the Hippo pathway activates. And so what we see is those are repressed in lupus, again, suggesting that the pathway, as we're defining it, is on, because we see that either in both non-lesional and lesional skin that you get decreased proliferation genes that are regulated by the Hippo pathway. And we saw this in both sets of data that we pulled into the manuscript.
And can you take that one step further for us and help us understand the functional impact of that differential gene expression in the keratinocytes?
We see increases in these pro-apoptotic genes and we see repression in these proliferation genes. So again, the cells look like they're being sort of primed to die if they get the right trigger. And so when we think about how lupus cells respond to UV light, that propensity to die is really important because it doesn't take much. You can look at them cross-eyed and then, off they go down the cell death pathway.
And it sounds like, and primed to apoptosis without kind of replenishment or without recovery, without proliferation in response.
Yes, exactly.
Got it. So the next series of experiments you did actually looked at that UVB-driven apoptosis, again, in the lupus keratinocytes. We know that lupus keratinocytes have increased apoptosis in response to UV light, and that's thought to drive photosensitivity responses in lupus, and particularly in cutaneous lupus. So your goal in that series of experiments was to investigate how Hippo signaling played a role in that. And you set up this quite complex series of experiments in which you overexpressed genes in the pathway and then used a couple of different mechanisms to downregulate expression. Could you talk us through those experiments?
Yeah, I'm happy to. So the first question we asked, I think, was sort of the obvious one based on the methylation data, in that WWC1 was hypomethylated, and we know this is that critical light switch for turning on, the hippo signaling. And so we just looked to see whether it was up or down in lupus skin at baseline, and we were able to see its expression was increased, which was exciting, suggesting that these methylation changes were really important, or were being seen in actuality in the cells in terms of increased protein expression. And so then we wanted to see whether overexpression of WWC1 was sufficient to increase apoptosis to UV light. And so we just made a cell line that overexpressed WWC1 stably. And what we saw was when we hit those cells with UV light, we could increase the number of cells that would die after a low dose of UV exposure. And then, lucky for us, we also had a fantastic collaborator, Dr. Ramiro Iglesias Bartolome from the NIH, who had also published a really beautiful tool that also could help us study this pathway. And he had generated a cell line, which fortuitously for us was in the exact same cell line that we were using to study the overexpression of WWC1. And there we could turn on an inhibitor that blocked a downstream interaction between YAP and TEAD. And what that did was mimic what was happening when you overexpressed WWC1. And so we could look at it and ask the same question in a different way to see if we got the same answer. And in fact, we did. So when we overexpressed this inhibitor that mimics the effects of WWC1 overexpression, functionally, we could again get increased apoptosis to UV. And then, further dug into dissecting the pathway, and there was a couple other tools that we had available. When WWC1 is overexpressed, one of the critical things that happens is a kinase called LATS gets activated, and there's two forms, LATS1 and LATS2. And so we wanted to see if LATS1 and LATS2 was also important. We showed that it was increased in its phosphorylation, both in the cells, but also I guess we didn't look directly in the skin, but we looked at the downstream substrate and that it was phosphorylated in lupus skin. But then if we inhibited the activation of LATS1 and 2, either by using a chemical inhibitor or by decreasing LATS1 and 2 expression using siRNA, we were also able to show that we could turn down the increased apoptosis that we see in lupus keratinocytes. So it took us from, modeling this in a cell culture system to then taking actual primary keratinocytes from healthy control in lupus patients. And so we could reverse the abnormality in lupus skin by blocking the LATS1/2 kinase.
Yeah, it's a beautiful experiment. And when I read it, I just was so impressed by what you did. And I will say, and for our listeners, there are a couple of kinase inhibitors that act on the LATS kinase, blocking the hippo pathway, that are under investigation for use in certain malignancies. Those agents are obviously in very early phase studies, but is it conceivable we might see a treatment beyond sunscreen coming over the horizon for patients with severe photosensitivity?
That would be awesome, right? So we actually got funding both from the NIH through a new R01 and also through the Lupus Research Alliance to continue to study this pathway. And we're actually looking at the impact of LATS inhibitors in a lupus model to see, one, if, how they act in a lupus model, if they impact disease overall, and then to study the photosensitivity aspect of it. So yeah, we're working on it, hopefully.
That would be brilliant. And I cannot wait to see the results of those experiments. I feel like that would be amazing for our patients and really change how we're treating lupus. So, Michelle, I wanted to take a few minutes now to talk about your life outside of work. Many people don't know that medicine is only one of your jobs. You are also a farmer, and you run Emma Acres Farm in Manchester, Michigan with your husband, where you raise heritage breed livestock, chickens, and organic vegetables. So I have to ask, Michelle, which came first, the chickens or the rheumatology?
You're so brilliant. That's wonderful. Well, probably the chickens. I mean, I grew up raising chickens. This is a nugget that I'm sure somebody will make fun of me for, but I was indeed the superintendent of the poultry department for our junior fair, 4-H fair growing up.
Fabulous.
The farm is a wonderful, and I have to confess, it's not run. Yeah, Mark, my husband, he runs the farm. He manages it. I help out where I can, provide some minor veterinary skills, and also I'm in charge of vegetables. So I run the garden and take 2 weeks of vacation in May to plant a huge organic garden, and we then sell vegetables and produce to complement the meat so that people can come to our farm and get everything they need to make an entire meal. So that's kind of a fun aspect of what we do.
I love that. And as the gardener in the family, in contrast to my garden, which I call my garden of growth opportunities, I think my garden has yielded about 3 beans this entire season, but your garden produces enough to both feed your family and other families in the region. What's your secret?
Well, I mean, again, like you say, it's you always learn from your opportunities. I definitely can't say that everything I've done has been a roaring success. And it's always a challenge when you're doing things organically. We adopt a no spray model. So we, embrace nature in its full aspects. And sometimes the fungus wants to take over, and you do what you can, but you have to acknowledge that sometimes Mother Nature has other ideas for your garden. But there's so much interesting data about, a bug bitten plant has more antioxidants than a plant that has no bug bites. And so I try to use that as a selling point for our vegetables when maybe they don't look as beautiful as you would like.
I think that's actually an important lesson. Ugly vegetables are always beautiful to taste. So how has farming life helped you grow, I guess, both as a physician and as a scientist?
Well, I think one, it's my own therapy, right? I call it my dirt therapy. It gives me an opportunity when you're doing physical labor to clear your mind and think of, I think, new ideas and problem solve. So I do use that as a way to exercise my brain in a different way. I think also one of the things that has become more apparent, I think, as we learn about our food supply and how nutrition and diet can impact health and immune health in patients, is that I've become very passionate about the importance of proper nutrition and healthy eating and avoidance of chemicals in our food supply. And so I think it sparks very interesting conversations with my patients in terms of how to modify their diet. And I think this is really a big area of need in lupus is what are the right diets, how does what people eat when they're young versus older, how does all of this impact disease, and how can we harness food to keep people as healthy as possible, I think are really important questions.
I completely agree. And I would love to know how you do that in a 20-minute follow-up visit.
Not always easy. Sometimes my visits go longer and I run very behind in my clinic, and my patients are usually pretty forgiving. We also have started at University of Michigan. One of our faculty, Aaron Stubbs, was a nutrition major in undergrad, and he's actually started a rheumatic disease nutrition clinic where he has dedicated half-hour visits to talk about diet and nutrition with patients. And that has been really awesome. He's building it out into an IRB-approved research tool where he can also conduct surveys and start to explore this with patients. So I think that's a really exciting thing that we're doing here.
Does sound exciting. I'm wondering whether you're including a pharmacy, F-A-R-M-C.
We've tried a few funding opportunities to get diet trials funded. So far, no luck, but we're going to keep trying. So
yeah, keep trying. I feel it's so needed. So my next question I ask of many of my guests, but you are someone who I'm really interested to know the answer to this question. You are someone who declined a speaking opportunity I offered you a few years ago. And so with so much going on in your life, how do you decide what to take on, what to give up? And do you have any tips for our listeners on the art of the polite decline?
Oh my gosh. Well, first, I apologize.
I think it was your son's birthday, so I think you had a great excuse.
Yeah. So when I try to figure out what the right balance is, sometimes you have to put family first, and there's certain things like birthdays that I prioritize. It's really hard. And I think especially as a woman in science, there's so many things you get asked to do because they want, gender equity on various committees and other things. And so I think sometimes women even get more burdened with these types of requests innately want to help. And so it becomes a real challenge. And I can't say that I'm very good at it. All I can say is that what I try to do is say yes to things that I'm passionate about and get involved in things that are going to increase my energy, not drain it. Also remember that when you take on something, you should offer your expertise, but then also look for opportunities on how you can give that opportunity to the next generation. And I think one of the things that we may not be as good at in rheumatology as we should be is pulling in younger people to leadership positions and elevating them to take over. And so that's one of the things I'm working on now, is trying to figure out, like, how do I bring more people up to do what I've been doing and let them put their own spin on it, right? So I think sponsoring and mentoring can be a way to, keep you from keeping things under your umbrella forever as well. So share the wealth, I guess. Yeah.
Good advice. The importance of succession planning, I think, is really important for every profession. You were also the first person who I heard describe the walk for women physicians through mid-career as walking through the valley of death. You have successfully made it out of the valley. Just a few months ago, you took on a new role at the University of Michigan as Vice Chair for Basic and Translational Research for the Department of Medicine. As you reflect on that journey through the valley, do you have any advice for those still walking through it? What survival skills did you rely on, and what did you wish you'd known or taken along with you on that journey?
Yeah, those are all really good questions. I think one important lesson I learned is that 2 + 2 = 5. And so building collaborative relationships as part of your science is really critical, because if you had to try to get all of your own funding and do everything on your own, I don't think I'd be out of the valley. I probably would have taken a left turn and ended up in LA or something. So I was very fortunate early in my career to meet Johan Gudjonsson, who's a dermatologist here, and we both had sort of landed on similar findings in sort of separate venues. And instead of competing, we decided to collaborate, and we, really went all in on our collaborative efforts. And I think I've benefited from his science and he's benefited from mine, and we've gotten some joint funding together, and we were able to establish a psoriasis and lupus cohort together. And so I think that finding good collaborators where you will get some of their grant money and they'll get some of yours, and you'll get on their pubs and you'll get on theirs. And, I think that type of relationship is really critical. And then also getting a really good peer support group, because, 90% of what we do is fail. We get rejected, we get rejected, we get rejected. And so having people who understand what you're going through is important. And I think for me, my husband is not in science. He's certainly smart and he has a master's degree and understands some things, but he's not going to be able to empathize in the same way as people who are dealing with rejections and reviews they might disagree with on a daily basis. And so, really having that support network that's broad, I think is also helpful just with the coping aspect. And then in terms of, what do I wish before I started the journey? I mean, I think I would have definitely loved more training in project management and personnel management, because I think the biggest challenges that I've had coming through science is really personnel. When you have challenging staff, students who have issues, other things that come up, I've sort of had to learn on the fly, and it would have, I think, probably been wise to have more training at the beginning. And then, just project management, as you get successful, you expand, and how do you keep everything going? And I think, again, I've learned a lot on the fly and I've learned by making mistakes, but I think, more training in that aspect, because a lot of what I do now isn't necessarily just what I was trained to do as a physician and a scientist. It's project management, it's budget management, it's public relations, it's, all these other things that, the skills are not necessarily emphasized early on. So
I'm wondering if some of those skills from the farm have been actually quite helpful in the kind of juggling of a huge translational research lab is kind of similar, right? You have all these different projects ongoing and things that need to be nurtured in different ways at different times, different seasons.
And then I think underlying it all is really just hard work. Like, you will never succeed in this field if you don't put your nose to the grindstone. And I think the farm definitely also brings that skill to the fore. And I can thank my parents for that installation of the appreciation of hard work from a young age. So
you got to be willing to get your hands dirty. That is for sure. Well, Michelle, I really wanted to say a huge thank you for taking the time to talk to us today. I was so excited when you didn't decline this invitation. I was thrilled that you were willing to share both your science and then your insights on life on the farm and life as a physician scientist. Thank you so much.
Thank you for having me.
Before we close today, I also want to give a shout out to our rheumatology spouses and significant others and supporters. Ours is a specialty that takes years of training and steadfast commitment, and our families and better halves often sacrifice to support that journey. Thank you for letting us do what we do. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow at ACRheum on social media. Email us at podcast@rheumatology.org and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.